Hi, and welcome to the UGA Marine Sciences virtual recruitment. My name is Kaylie Plum, and I'm a master's student under the advisement of Brian Hopkinson, and I study biological oceanography. So I grew up in Rockford, Illinois, really far away from the ocean, but I always knew I wanted to study marine science. So after graduating high school, I decided to attend the University of South Carolina, go Gamecocks, where I studied marine science with a concentration in biological oceanography. While I was there, I spent a summer doing a research fellowship on the coast of South Carolina, where I got to analyze a decades-long time series looking at zooplankton, community composition, and also contributing to sampling every week to keep that time series going. And it was a really cool experience that helped me decide to continue going on with my education and keep getting research experience. So once it was time to graduate from my undergrad, I decided to continue on to grad school here at UGA. So that brings me to my current work here. I decided kind of to switch gears and work with phytoplankton instead of zooplankton in graduate school. So what I'm doing now is working on a project looking at the use of this protein, proteorhodopsin, in diatoms. So proteorhodopsin is a protein that's mostly used in heterotrophic organisms to harvest some energy from the sun to supplement their diets. So it's pretty strange to see it in diatoms since they're already able to harvest sunlight through conventional photosynthesis. So the hypothesis is that diatoms use proteorhodopsin to make energy when they're iron limited and aren't able to photosynthesize quite as well. So looking at the upper diagram, you can see how much iron is required in almost every structure of the system, whereas proteorhodopsin doesn't require any iron to function at all. So that brings me to the goal of this project, which is to measure the energy flux through the conventional photosynthetic system and compare it to the energy flux through this proteorhodopsin. And we want to do that under iron limited conditions and also when the diatoms have plenty of iron to function. And we want to see if the proportion between energy moving through proteorhodopsin versus photosynthesis changes at all under those two conditions. So I haven't quite finished yet. But I have been able to analyze the changes in photosynthetic flow versus in a iron replete diatom versus an iron limited diatom. And so far we're seeing what we would expect. The blue arrows and blue text represent stages of photosynthesis that significantly decreased under iron limitation compared to the iron replete sample. So hopefully in the next few weeks as I finish up, I'll be able to start filling in these numbers here. And that's going to give us a better idea of how large of a role proteorhodopsin is playing in these diatoms. Lastly, I just want to talk about why I chose to come to UGA. Besides just having the opportunity to work on this project here with Brian, I also chose UGA because of its location. For me, Athens is a great city to spend grad school in. It's not too big, it's not too small. It's pretty affordable to live here. And outside of a pandemic, there's always fun and interesting things going on in the city. Also, the department itself just has a really great group of grad students. We've got a grad student association. So there's always monthly social events and outreach events going on. It's a little bit different during the pandemic, but there's still that effort there to get together and stay connected, just like when things are normal. Also, there's just a lot of interesting research happening here at the department. They have a nice series of courses called synthesis skills where you can learn important skills for scientists to have that you might not learn in a general oceanography class. So those have been really useful for me to develop as a scientist. And yeah, overall, I've had a great time here at UGA, and I hope that anyone attending this recruitment event will come in the fall. And if anyone has any questions about my research or Brian's lab or UGA in general, feel free to contact me. You can email me here. And yeah, I hope this was helpful.